This video is to help you understand and practice some of the concepts that go along with the mathematics and information and communication technology skills. In biology, we're going to be collecting tons of data, okay? That's the easy part. The hard part is trying to understand what exactly you're supposed to do with it. And there are several different ways that we can analyze that data. One of the ones we're going to be using most often is to find the mean. Okay, the mean is also the average, and to find that, you're going to take the sum of all of your numbers and you're going to divide by the number of numbers. Or if you're clever, you're going to make your spreadsheet do that for you. And that's going to tell us the average or the central tendency of data. One of the things that you'll never have to do for me, but you might be reading about in some data, is finding the median. The median is the middle number, and to do that, you would have to take all of your data points and put them in numerical order and then find the center one. I think that's a task that teachers give their students to make them be quiet. You'll never be doing that for me, but if we're interpreting a median, what that tells us is that's the number that separates the higher and lower halves of the data. And it's really good if we have outliers. So for this set of data down at the bottom, I have teacher ages, and we can see just by looking at them that a lot of the ages tend to be really close together, and then I have this one outlier over here. If I were to use the mean, then this outlier would kind of push my mean a little towards that number more so than it really should in order to be representative. So for something like this, we would want to use the median, okay, the middle number, and that might give us a more representative value. Another way we can analyze data is by finding the mode. That's the number that occurs the most often. It tells you the most common result. Okay, so you can see which things appear with the greatest frequency. And then the range. The range is the highest minus the lowest value. So the difference between the least and the highest value that you have in your data set. It is only one of the ways to measure the spread of data, and it's not the one that we're going to be using most often. We'll get to that one in just a couple of minutes. One of the most common things to do with these super large data sets is to put them into a graph to give us a better visual representation of what our data is trying to tell us. So here we're looking at what I can assume is the average number of tornadoes per month. Again, this is not a very good not a very good graph, because it should say that. And this tells us that some months have higher average numbers of tornadoes than other months. What it doesn't tell us, though, is which months vary more than others. So for example, are there always 5 tornadoes in January, or does it range from 0 to 10? Are there always close to 400 tornadoes in May, or some years are there 800 and some years are there 200? Okay, so this gives us some good information, but it doesn't tell us anything about the spread of the data during those months. To incorporate that kind of information into your graph, you're going to be adding something called an error bar. Now, this is kind of a crappy name for something because errors kind of have a bad connotation. They're not necessarily errors like you did something wrong, okay? These bars represent the variability in the data. Okay, so they tell us how big of a spread those data points are. Error bars can either represent the range or something called standard deviation. Don't worry if you don't know what that is yet. We'll get to that in a little bit. So in this graph, I'm looking at the mean cell size of onion cells and cheek cells. Judging on where the bars are, I can see that onion cells have a higher mean than cheek cells. But because the error bar is smaller, okay, this tells me that this one has less variation in the data, and this one has more variation. So in other words, okay, the ranges or the standard deviations of cheek cells okay, are much greater. They come in many more different sizes than onion cells, which are a little closer together. So I just told you that error bars can represent the range of the data, the highest minus the lowest, but most often we're going to be using error bars to represent something called standard deviation. Now, standard deviation is actually a number, okay? It's a numerical value. If you really have nothing better to do, you can calculate it by hand. I'm going to show you later how to calculate it on a spreadsheet, but in the meantime, we just need to have a basic understanding of what this is. And standard deviation is a numerical way to represent data variability. So in other words, how close the values are to the mean. Are they tightly packed together around the mean, or are they all spread out all over the place? What we're looking at here down in this graph are two different values, okay? The red graph shows a lot of data points really close to the mean, and the blue graph shows a mean over here, but the data is kind of spread out all over the place. The higher the standard deviation, okay, remember standard deviation is a number, the more spread out the data is. So I'm looking over here, it says the blue has a standard deviation of 50. That's pretty high, okay? So that means that they're very spread out. The red graph has a standard deviation of 10. It's a much smaller number, meaning that the values are a lot closer to the mean, a lot more tightly packed together, whereas whatever data they gathered and graphed in blue is much more spread out. Now, for our purposes, we're always going to assume that our data is normally distributed around the mean. So what does that mean? Well, it means that on either side of our mean, I'm going to find half of my values and the other half are going to be over here. We're going to make that assumption because it makes our mathematical lives easier. Is that always the case? Obviously not, but we're going to make that assumption for now. So let's take a closer look at this graph. Whenever I'm doing standard deviation problems, I always draw a graph like this and I label it to help understand what's going on with different numerical values. Okay, so in the middle here, okay, at our peak, this is the mean. Okay, so again, this is our mean value. This x-axis is some kind of number, so if these are test scores, okay, these would be the test scores, and this would be the frequency, so how many students scored that particular score on a test, okay? So here's some kind of value, up here is frequency. Right in the middle here we have our mean. Now, we said that standard deviation is a number. What does that number mean? Well, in order to kind of figure that out, I'm going to label what +1 standard deviation, so 1 standard deviation, I'm going to abbreviate, 1 standard deviation above the mean, that's why I used +1, and 1 standard deviation below the mean, so -1 standard deviation, okay, falls there. Now, the rule is that 1 standard deviation above or below, so plus or minus 1 standard deviation, is going to account for 68% of your data. So what does that mean? Well, that means that 68% of the students, if these are test scores, scored in between whatever those two scores are. Okay, so again, if you know the number that's the mean and you calculate the standard deviation and you add 1 to the mean to get out here and you subtract your standard deviation value to find this value out here, what that tells you is that 68% of all of your data points fall within these two numbers. Now, you could keep going for an infinite number of standard deviations. We're going to go one more. Okay, so I'm going to label +2 standard deviations. So I'm going to put on a second standard deviation and take away a second standard deviation. Okay, and that's going to represent a different percentage of my data. So between these 2 points, okay, that's going to encompass 95% of my data points. So plus or minus 2 standard deviations represents 95% of the data. So again, if you know the mean, and I'm going to write this down, you don't have to do this because we're going to practice this about 1,000,000 times, okay? But if I'm doing an example here and I say, okay, the mean test score was 80 and the standard deviation equals 5. Again, that's a mathematically calculated value. Then one standard deviation above the mean would be 85, the mean plus a standard deviation. 2 standard deviations above the mean would be a 90. One standard deviation below would be 80 minus 5, which is 75, and 2 standard deviations below would be a 70. Okay, so what do all these numbers mean? Well, that means that the mean was 80. That means 68% of the students in my class scored between a 75 and an 85. They scored between 1 standard deviation above and 1 standard deviation below. 95% of my students scored between a 70 and a 90. Okay, scored somewhere in here. So you're going to get a lot of questions about a graph like this. Don't be scared if questions start to arise that only ask for 1 standard deviation above or 1 standard deviation below. It's okay. The mean splits all of these values in half. Okay, so this 68% here, half of it, and you don't have to write this, but you can if you want, you don't have to. Half of this 68% is here, and the other half is here. So 34%, half of 68, is 1 standard deviation above. 34% is 1 standard deviation below. Okay, and that can apply to 95 as well. Okay, so this is one of the practice problems in your packet. It's not in your packet. It's just to practice to make sure you and I go over how to do these things. All right, and again, I always start off with drawing a graph. So this says I have 120 students and the average test score was 87, the standard deviation was 6. All right, so I always start off by drawing one of those graphs. Okay, and right here is my mean. It says my mean was 87 and my standard deviation was 6. Okay, so I'm going to draw lines for +1 standard deviation, -1 standard deviation. Okay, and each time if I want to figure out what those values are, since it says my standard deviation was 6, 87 plus 6 would be 93. 87 minus 6 would be 81. Now I'm going to draw lines for +2 standard deviations and -2. I'm going to add another 6. So that would get me up to 99 and 75. Okay, so now let's answer some questions. First question, what percentage of students scored between a 75 and a 99? Okay, 75 and 99 is here to here. So that's within +2 or -2 of the mean. And we know that +2 or -2 standard deviations from the mean incorporates 95% of the data. That's just the rule. Okay, now this question's a little trickier. It says how many students scored below a 75? Okay, below a 75 is represented by this tiny portion of our graph over here. And there are many different ways to figure this out. Bravo to you if you want to do it differently than me, as long as we get the same answer. So if I know 95% of my data goes to here and here, that means I have 5% of my data left unaccounted for. half of it is going to be down here and half of it is going to be up here, okay, of this 5% that's left over. So that gives me 2.5% up here and 2.5% down there. So the answer to what percentage of students scored below a 75 would be 2.5%. But it's not asking me the percent, it's asking me how many. So I need to figure out what 2.5% of 120 is. Again, several ways you can do that. I tend to turn percentages into a decimal by moving the decimal over 2 spots. Okay, so that would give me 0.025. That's how we write 2.5% as a decimal. And then I would multiply that by my number of students. And I should have pulled out a calculator before I started this video so that I could tell you this on time without having this awkward pause here. But here we go, 0.025 times 120 gives me 3 students. Let's do another practice before you have to do things on your own. This one says a certain basketball player scores an average of 24 points per game. Okay, so here's my graph. 24 is going to be right here in the middle. Let's assume that he scored between 15 and 33 points for 68% of the games he played. Well, we know 68% is plus or minus 1 standard deviation, because that's the rule. Okay, so when we say 15 and 33, that means that those are our standard deviation values. So out here at +1, whoops, that's not a plus on that side. Out here at -1 standard deviation or +1 standard deviation, okay, that's where those 15 and 33 values come in. Again, and I know that because 68% of the rule, or 68% is the rule for plus or minus 1 standard deviation. Now I can assume what the standard deviation is. The standard deviation has to be the difference between the mean and whatever our +1 or -1 values are. And so the difference between 24 and 33 is 9. And that works out on this side too. So now I can go ahead and label the +2 and -2 standard deviation values. So I would add another 9, which would be 42. I would take away 9 here, which would be 6. And so now I'm ready to answer some questions. This first one says, what percentage of the games ended up with him scoring 42 points or higher? Okay, well 42 points or higher is this area out here. And again, I know that 1 standard deviation is 68% of the value, or I should say the values. 2 standard deviations is 95% of the value, okay, meaning I have 5% left. Okay, 5% is going to be equally distributed on this side and on this side. So 2.5% is going to be out here, and another 2.5% is going to be over here. So what percentage of the games ended with him scoring 42 points or higher? That should be 2.5%. If he played 81 games, during how many games did he score less than 15 points? Okay, now this one is a little bit tricky. So less than 15 points is this area out here. So all of this chunk. All right, the easiest way to do that is to kind of figure out what we have left to work with. I know that out of this 68%, this area in here, 34% of it is going to be over here, and the other 34% is over here. So that's another way of writing that. I also know that out of my whole graph, half of all of my values fall on this side, and the other half would fall over on this side. So here's what I want to know. Well, here's what I know is 50%. Okay. Here's what I don't need. This area in here, what I don't need, I know is 34%. So if I take 50 minus that 34% that I don't need, I know that I'm left with 16%, and that's what would be in here. So that should be 16%. Again, though, that's not what it's asking us. It's asking us how many games, not what percent of games. So I would have to turn 16% into a decimal, 0.16, and then I would multiply that by the number of games, so 81, and 0.16 times 81 gives me 12.96. I'm going to round that to 13 since you can't play a decimal of a game. At this point, you should be pausing the video and completing numbers 6 through 8 on your own. When I look at your packet, I should see work like graphs, multiplication, etc. So pause it, solve these, and then I'll show you the answers so that you can check your answers. Okay, here come the answers. So I hope you're taking just a moment to check these. If you're finding that you're having difficulty, please just make a mark next to them and we can talk about them later. Okay, so without actually calculating standard deviation, we can sometimes look at data and just know. Okay, so here I'm looking at test scores from 9th graders and from 10th graders. They have very similar averages, but very dissimilar standard deviations. These are all identical. Okay, they're all the same as the mean, very tightly grouped around the mean. These values vary a lot more, so they have a lot more range, they're not as close to the mean. So my group of 10th graders has a larger standard deviation. And we can see that if we make spreadsheets actually calculate it for us, what the standard deviation there would be. Now, we do have different means. The 9th graders had a higher average than the 10th graders. But can we confidently say that 9th graders have better test scores than 10th graders? Well, in order to be able to answer that question, we have to perform an actual statistical test. Because see, when we're just looking at data, okay, and we're just looking at the means, we can tell that they're different, but we don't know if they're significantly different because means don't include any kind of information about variability. If I have a lot of variability in my data, maybe my experimental design isn't very good, maybe it's not taking all the factors into account, okay? Maybe I have a small sample size and my 10th graders were all sick that day. I don't know, okay? So I need to be able to interpret these values, keeping means and variability in mind, okay? How much or how consistent those data points are in order to be able to tell if they're actually different. Now, before I tell you about the statistical test that we're going to use for that, there's a visual test that you can use also. So remember, we said that one of the things that error bars can represent is standard deviation, okay? Large error bars mean that you have a high standard deviation, and that means you have high variability, that your values aren't very close to your mean. When I'm looking at a graph, if those error bars overlap, and if those error bars represent standard deviation, that means that they probably have a lot of data points in common. Remember that if we're using these to represent standard deviation, one standard deviation is 68% of my data. So 68% of my data falls in here. 68% of this data falls in here. So even though they have slightly different means, the fact that their error bars overlap means that they share a lot of common data points, and they're probably not significantly different. Okay, so if the error bars overlap, they're usually not significantly different. If error bars do not overlap, so I'm guessing this error bar doesn't overlap with this error bar all the way over here, that means that those two values probably are significantly different. Okay, so the more your error bars overlap, the more data points you have in common, and the less likely it is that those values are actually significantly different. If you don't have overlap, that means it's more likely that they are significantly different, which means that we can confidently say that those means are actually different. So again, that's a little bit easier to determine when we have a graph with error bars that we're looking at. If we're just looking at data, it's a little bit harder to tell. So for that, we use a mathematical test. And that test is called the t-test. Okay. The t-test is used to determine the significance of the difference between two means. So it tells me, is the mean for my 9th graders significantly different than the means for my 10th graders? Always takes into account two means. When we calculate it, and again, we're going to be calculating this using spreadsheets, so you'll never have to do this by hand, but it takes into account the means of the two groups and then other factors that we tend to forget about, including the standard deviation or variability, and the sample sizes also play a part as well. Now, I'm going to teach you how to interpret the actual mathematical results of the t-test, but just something to kind of keep in mind here is that the t-test is probably going to show that two means that are really close together, or two sample sizes that have lots of variability probably aren't going to be significantly different, okay? So let's take a look at how these t-tests are calculated. So the next part of a t-test involves something called a null hypothesis, and I know a lot of you are into the naughty habit of thinking that a hypothesis is an educated guess. Get rid of that notion. That does you no good. It tells you nothing useful. Okay, so we're going to be a little more specific. A null hypothesis is written, that's kind of not even how you write it. You write it as H subscript 0. Okay, and it's the big, like, Debbie Downer of science. The null hypothesis is always that there is no significant difference. So no matter what your experiment may be, you state the null hypothesis as there is no significant difference. And the t-test is either going to support that or refute it. Okay, so if I'm looking at this data, again, the null hypothesis would be there is no significant difference between the scores of 9th graders and 10th graders. And we're going to use the t-test to figure out if we should accept that or reject that hypothesis. So what is a t-test? Okay, well, a t-test, okay, compares two means, but when you use something like numbers or Excel to conduct a t-test, what comes out of the t-test is the p-value. So a t-test will generate a p-value. And p stands for probability. It's the probability that any difference in the means is only due to chance. So if p is very close to 1, if p is a high number, that means that there's a high probability that any difference in those means is solely due to chance, okay, and not to some actual systematic difference. If we have a very small p-value, like very close to 0, that means that there's a low probability that that the difference is due to chance, and that it's much more likely that the difference is actually due to some kind of difference in your system, like a difference in 9th graders and 10th graders, and not just due to chance. If something has a p-value of 0.5, that means 50%. So 50-50 chance that whatever kind of difference is between your means, that the probability is just due to chance. In biology, we use a standard p-value of 0.05, okay, to determine the significant difference. If p is less than 0.05, we can confidently say that the means are significantly different, and we can reject the null hypothesis. If p is greater than 0.05, we have to accept the null hypothesis because that means that the means are not significantly different. So I tend to think more in terms of tables. So for me, instead of writing all this down, I would kind of write and fill in a table, but that's totally up to you how you want to do that. But just to review, okay, if P is less than 0.05, then yes, they are significantly different, and we get to reject the null hypothesis. Remember, the null hypothesis says that they aren't different. A p-value of less than 0.05 would cause us to reject that. If we get a p-value of greater than 0.05, that means that no, we cannot confidently say that they are significantly different, and we are forced to accept the null hypothesis. So back to our 9th grade and 10th grade data, okay, I took this data and ran a t-test, and I got a p-value of 0.86. Okay, again, if we're using our P as 0.05 or less benchmark, okay, then this means that they are not significantly different, and we have to accept the null hypothesis. So sometimes we're not comparing two groups, okay? We're not always going to say, is this group different from that group? Is this way different from that way? Sometimes we're going to be collecting continuous data, okay? And we're determining if there's a relationship. So maybe, is there a relationship between foot length and height? Okay, well, in order to do that, I would collect data on people's foot length and their height, and I would plot them in a graph, okay? And then I might add a trend line or something like that, and I can see here that there's a pattern. So in general, as someone's foot length goes up, so does their height. And it's easy to kind of slip into this notion of saying that having a larger foot causes someone to be higher. We have to be very careful about using that word cause. Because correlation, a relationship in variables, does not mean causation. So in other words, two factors may follow a similar pattern, but one doesn't necessarily cause the other. So here I can see a very tight correlation, a very strong relationship where they follow the same patterns between the number of fresh lemons imported and highway fatalities. They may follow the same pattern, but that does not mean that that importing more lemons causes a lower highway fatality rate. It does not mean that one causes the other. If you wanted to take your correlation to the next level and see if one variable actually causes another, then you need to do controlled experiments, okay? You would need to, in this case, find some kind of sample where you have people and you can measure how long they've been smoking, and eliminate every other variable. So eliminate differences in genetics, eliminate differences in lifestyle, eliminate differences in environment, okay? And then calculate their cumulative risk of lung cancer. You have to do controlled experiments where you're isolating only one variable in order to determine if one thing causes another. So be really careful when we're looking at things in the news. Graphs like this pop up a lot. This graph is even in your textbook somewhere. We can say that the age at which smoking stops is definitely correlated to the cumulative risk of lung cancer, but we cannot say that one causes the other until we've done a controlled experiment. And as you're going to find out with humans, that's very, very difficult to do because there are so many different factors and ethical concerns. So we mainly do them with animal studies, but again, has to be a controlled experiment. Okay, so throughout this video, I've shown you several different types of graphs. It's kind of time to go back and talk about why I've chosen to show you certain types of graph in different situations. Different types of data and the way that we analyze that data require different types of graphs, and we'll go over that later. It doesn't matter what type of graph you're using, they all should have some similar characteristics. They should all have descriptive titles. Okay, so our titles, if we're graphing average data, you need to say that those are averages. Okay, be specific in terms of your variables. Okay, make sure that you're defining what time periods you're using. Make sure you're defining where you are. If you're including units here, that's fine too. If you have error bars, we'll get to that, okay? But your titles need to be very specific. You also need to have correct X and Y axis labels. Okay, so down here on the x-axis, okay, these are labeled here, year. If this were something else like absorbency, let's say, you would also want to have units in there. And we also need to have very clear data labels. Okay, so if we can, we want to make sure that we have data labels. If it's not possible, make sure that your axes are labeled in a way that it makes it easy to kind of discern what values we're looking at. If your graph includes error bars, okay, and it will, anytime you are doing a bar graph and anytime you are graphing averages, we also want to include error bars. I'll show you later on how to add that, but since error bars can represent range or standard deviation, you need to state what they represent in your title. You must say what those error bars are. When we're making graphs, we're generally going to use one of 4 major types of graphs, and this is going to depend on what kind of data we want to display. So the first and maybe one of the most common types we're going to use are bar charts. And bar charts are used to show the amount of data in certain categories. They generally look something like, I don't know, like this. Okay, something like that. Usually we put error bars on them, okay, but not always. Anyway, so they look like something like this, and they show different data in different categories. So here I gave the example of preventable causes of death. There are different categories. Notice that these are categories and not values of numbers. Okay, so they show us the frequency of data in different categories. This would also work if I graphed like average height of boys and girls, because boys and girls are categories like these things, not values. If I needed to show the frequency of data that appears in certain value ranges, then I would use a histogram. Okay, histograms look more like this. Well, first of all, the bars are connected together, and they're connected together because each bar is for a range of continuous data. So instead of the last bar graph like we looked at that had like smoking, car accidents, cancer, whatever, okay, histograms show the number of values or the frequency that fall between 2 ranges, so between 60 and 65, 65 and 70. And because those ranges are continuous, okay, those bars touch each other. So down here we can see the difference between histograms and bar charts, okay, categories versus number ranges, gaps versus no gaps. So really for our purpose, it's more important to understand what's down here, because these help us to decide what type of graph we're making based on the kind of data that we have. Another common type of graph is the line graph, and line graphs are used to show how things change over regular increments. Okay, so down here we have individuals per 1,000 and maternal age. Okay, so tracing the incidence of Down syndrome through maternal age. We have a continuous range of maternal ages down here, and so these are averages for certain ages. So line graphs are always going to look something like this, okay? And it's important to note that for line graphs, we connect the dots. Okay, so maybe I'm going to make a note here that the dots or the data points are connected with a solid line. Okay, so that's actually what it means to be a line graph. So we connect those points with a solid line because it's average, 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 average. Okay, so these are actual relationships. And that's a lot different than our last type of graph we're going to talk about, which is the scatter plot. Scatter plots don't track averages over single increments. Scatter plots, when we were making them, we're actually plotting all of our data. So maybe it's important to mention that for line graphs, usually we're plotting an average, where when we're plotting using a scatter plot, we're usually plotting all of our data points. And how do we know that we need a scatter plot? Well, this is when we may have more than one y value for a single x value. So if I took a sample of 100 adults and I plotted their weight and their height together, it's totally possible to have two adults that weigh the same but have different heights. And so it's important here that I represent all the individuals in my data study and that I plot them in a scatter plot rather than a line plot. Notice that these dots aren't connected. Okay, if I wanted to show a general trend, I could add what's called a trend line. We'll talk more about that later. But the points aren't connected with a single line like they were in a line graph. So maybe back when you were in like the 6th grade, you did some scatter plots, and then maybe you got out a ruler and you drew a line of best fit or a trend line. Well, get with it. Those days are gone. We don't draw those things anymore. We let computers do that for us. We let our graphing software take care of that. Because these trend lines are actually a mathematically calculated line. They're not a guess. Okay, these lines are made in such a way that all the dots are an average distance away from this line. So there's just as many data points above the line as there is below the line, and the average distance of the dots from above the line is the same as the average distance of dots from below the line. So it's all very mathematical. We're not guessing at these anymore. And this trend line is here to show a general pattern, and more importantly, to allow us to predict a y value for a given x value. So if I draw in a trend line here, it's giving me a general idea as to the trend in the data. I can calculate a slope or a rate. But what makes trend lines pretty cool is they allow us to predict y values for x values that maybe we didn't test. So it doesn't look like in this study that anybody that weighed 80 kilograms, I hope this isn't kilograms. Well, no, sorry. Those aren't height and weight. Those are literacy rates. Sorry. We didn't have a country that participated in anything that had an 80% adult literacy rate. So let's say I discovered a new country and it had an 80% adult literacy rate, and I wanted to just make a guess or a prediction about the life expectancy of people there. I could use this trend line to make a guess or a prediction and say, okay, well, probably those people are going to live around 64 years. Okay, so trend lines are really cool in that way. They allow us to predict y variables based on x variables that maybe we did or did not test. Now, some of these trend lines or lines of best fit are better at predicting y values than others. Okay, so if my data really kind of clusters around this line and really kind of holds true to this prediction, then this line is probably a pretty good indicator, okay, of where those data points might be. If my data is highly variable, okay, like it's not really all that predictable, and I get a trend line, that trend line may or may not be a very accurate predictor. So according to this trend line, okay, any country that has a male to female literacy gap of close to, like, let's say 25 is going to have an urban adult HIV rate of around 5, well, that's a little bit more than 5%. Okay? So that's what this line is telling me. But my data is so variable. Look, for things that are very close to that literacy gap, I'm all the way up here. Okay, I'm all the way down here. This line is not very good. Okay? Now, how can we assign a number to how good that line is at predicting things? Well, we have a number for that, of course, called the correlation coefficient, okay, or R squared. It's a number between 0 and 1 that tells us how well the trend line matches the data collected. Is it really tightly clustered? Does that line do a really good job of predicting values? Or is that data highly variable and that line may or may not be a good predictor of values? So here's how we're going to determine or interpret that R squared value. The closer to 1 that value is, the better the trend line fits the data and the better predictor that line is. Okay, so a better fit and a better predictor. Where are we getting these values from? Well, we're going to make our spreadsheet software calculate that for us. We're going to practice that, don't worry. But when you graph your data down here, okay, there's a way to make the graphing software add in a line of best fit or a trend line, and it will also calculate your R squared or your correlation coefficient for you, and it's going to give you some kind of a number. So again, the closer to 1 it is, the better that line fits your data. So here my line of best fit would be right on all these points, and the less variability there is in your data. Okay, as we go down, get smaller than 1, okay, we're still here at 0.8, that's pretty good. 0.3, these dots are really starting to get scattered. And 0, there's really no correlation here. So the closer to 0 we are, okay, the poorer their fit is and the less reliable that trend line is at predicting the data. Okay, now we have a couple of hard and steady rules for how we talk about this. If we have an R value of greater than 0.8, so something like one of these, then we say that's a strong correlation. Again, not causation. We can't say that. Okay, but we can say that that is a strong correlation, that whatever this variable is strongly correlated to this in such a way that most of the data points seem to follow the trend of that line. If we have a value of less than 0.8, that's what we call a weak correlation. That, okay, yes, when this changes in the same general pattern, but not so much so that I can use a line to accurately predict data, that the data is kind of all over the place. And if we have a value of 0, then we have no correlation, that a change in this number doesn't seem to cause a predictable change in this number. There's no pattern. Okay, so far we've talked about a couple of different ways that we can analyze data. We talked about t-tests, which test the difference between two means. We can talk about R-square value to see how closely correlated things are. We've talked about standard deviation to talk about how variable our data is. One last type of test that we really need to go over is called the chi-squared test. Okay, instead of seeing it spelled out like this, sometimes you may also see it like this, okay? A capital X or a chi squared. And the chi-squared test is used when we want to see if our predicted results significantly differ from what we expected. Okay, so just to contrast that with our t-test, remember the t-test is the difference in two means that you measure. Okay, so are boys taller than girls? Do ninth graders score better than 10th graders? Those are both things that we actually measured. I measured ninth grade scores. I measured 10th grade scores. Or I measured girl heights versus I measured boy heights. The chi-squared test is used when we want to see if our measured results differ from what we expected. Okay, and so this takes into account, again, your observed values, what you measured, your expected values, okay, and we're always going to be working, this is important to note, in numbers, not percentages, okay, because when we work in numbers, that inadvertently or indirectly takes into account sample size also. So just like with the t-test, we're going to start with something called the null hypothesis, or H subscript 0, that there is no significant difference, okay? So that in the t-test we had a null hypothesis that said there's no significant difference between the two means of data that we measured. Here, for the chi-square test, the null hypothesis is that there is no significant difference between what we expected and what we actually observed or measured. So in other words, if we do find a difference, it's probably just due to chance or random sampling errors. Okay, so teaching you how to do a chi-squared test is a lot easier if we just work through a problem together, okay? And so the first thing you want to do in a chi-squared test is to actually take your measurements, get some data, okay? So let's pretend that I do a sample of 500 people. I look at 500 people in McCaskey High School, and I find that 100 of them have blue eyes and 400 of them have brown eyes. Okay, so what? What I want to know if I'm doing a chi-square test is that different than what we expected? Well, to understand that, we really have to go back and think about what did we expect? So here's where things like Punnett squares come in, and we're going to talk a lot about these later in the year, okay? But if I do a Punnett square from two heterozygous individuals, meaning one allele or one gene for each of them, then what this Punnett square is telling me is that 1, 2, 3 out of 4, or 75% of my people should have brown eyes, okay? And 1 out of 4, or 25%, should have blue. So let's figure out if what we observed is significantly different than what we expected. Okay, so first I'm going to write down my observed numbers. When I'm doing chi-squares, I draw a chart like this, always with these headings. These are based on how many outcomes there are. In this case, there was only 2 outcomes, brown or blue. If you were doing something with what month were you born in, then you would have 12 outcomes. January, February, March, April, May, June, July, August, September, October, November, December. Okay, but anyways, I start with a chart like this. So first step, measure the observed numbers in each category, and I'm going to write those in. So let's say this column is for brown, and this one is for blue. My observed numbers I'm getting from up here. I found that at McCaskey, okay, 400 people had brown eyes and 100 people had blue eyes. I'm not going to worry about any of these sum columns. Okay, we'll do that at the end. The next row is for our expected numbers. And I know over here my expected percentages, but they have to be in numbers. So to find these, I'm going to take into account my expected percentages. So 75% is 0.75 as a decimal. And if I want to find out what 75% of 500 people is, I would take 0.75 times 500, and that gives me 375. Okay, with blue, I'm expecting 25% of my people to have blue eyes, so that's 0.25 times my sample size of 500, and that gives me 125. Okay, so I have my observed values, I have my expected values. Now I need to find the difference, which is going to be O minus E. Okay, observed minus expected. 400 minus 375 gives me 25. Okay. 100 minus 125 gives me negative 25. Now we're going to find our difference squared. So observed minus expected, which is what we found up above. Now we're going to square it. Okay? So 25 squared equals 625. Minus 25 squared also equals 625. And now the last step, well, one of the last steps, is I need to take my difference squared, so these two, okay, and I'm going to divide by the expected value. So that would mean I need to take 625 divided by 375. So this, observed minus expected squared, divided by my expected. And here that gives me 1.67. Over here, I would be taking 625 divided by 125. And that calculation gives me the number 5. The last thing I need to do to find chi-squared is to add all of these. Okay, well here there's only 2. Okay, but I do need to find the sum of these two numbers. And that gives me a chi-squared value of, well, it'd be helpful if I did this right, of 6.67. So what the heck does that mean? Okay, well, we have to go one extra step here. So keep in mind this chi-squared value of 6.67. Because we're going to have to look that up in a table in just a moment. Okay, so I'm going to just write this down again, that for us, chi-squared value was 6.67. So the same rules and applications that we learned from the t-test apply here too. We're going to be looking for p-values of less than 0.05. So if p is less than 0.05, it is significantly different, and we can reject the null hypothesis. If it's greater than 0.05, we have to accept the null hypothesis that they are not significantly different. But unfortunately, we're not going to have the benefit of looking these up or getting numbers to just generate these values for us. We have to look them up in a table. And tables always have the probability over here. And again, we're only concerned, if P is probability, we're only concerned with this column, P equals 0.05. That's the only one we care about. And then over here we have something called degrees of freedom. I don't know, it sounds like some kind of hippie rock band, but it's really a statistical variable. And so how do we get degrees of freedom? Well, degrees of freedom are calculated by taking the number of different outcomes minus 1. So in our example, we had 2 outcomes, brown and blue. And then to find our degree of freedom, we're going to take this minus 1. And that gives me a degrees of freedom of 1. So to find out if our chi-square value means if we had a significantly different result, we're going to cross-reference the degrees of freedom, which in our case was 1, with our p equals 0.05 column. So I'm going to follow this all the way over here, and I'm going to get to this value, 3.84. What the heck is that? Okay, now it's time to go back to our calculated chi-square value. If our calculated chi-square value is greater or bigger than what we just cross-referenced in the table, then there is a significant difference between our observed and our expected values. So in our case, the chi-squared value that we worked so hard to calculate is greater than this cross-referenced value in the table, which we found with degrees of freedom and the p-value. Okay, this is greater than this. So that means that there is a significant difference between what we observed by looking at people at McCaskey and what we expected using that Punnett square. So just to kind of summarize how to interpret these table values from the chi-squared test. If the calculated chi-squared, so what you found in your calculations, is less than what's in the table, then we have to accept the null hypothesis that there is no significant difference and that any difference in what you observed versus what you expected was probably due to random chance. Okay, bad samples, small samples, variables, all that kind of thing. If what you calculated as your chi-squared value is greater than or more than what was in that table, then we reject the null hypothesis because there is a significant difference and there's an actual difference between the samples that you observed and what you expected of them. So maybe the predictor model, like the Punnett square that you were using, really doesn't apply to that situation. So it's a useful test in that way. Okay, so if your brain's like mine, you're tired of numbers for the moment. Let's take a break from that and let's go back and just review a couple of other important random tidbits of things that you need to know that aren't necessarily in your book. And one of those is the difference between qualitative and quantitative data. When we're collecting data from a lab, we may be focusing on one more than the other. We may be looking for color changes, or we may be measuring things, but we need to keep in mind and not forget the other type of data also. We should always be collecting both types of data in a lab. So qualitative data are observations, okay, using one of your 5 senses. It turned color. It made bubbles. It smelled like rubber. It felt bumpy. Okay, so things that we can't actually assign a number to. Whereas quantitative data is exactly what it sounds. It has to do with the quantity. So here our answers are always going to include numbers because they come from calculations or measurements. Another important thing that I can't believe your book leaves out are our different kinds of variables. Okay, so we have an independent, a dependent, and then controlled variables. The independent variable is what you think might be responsible for a certain result. So if I want to see if the color of light makes a difference in the average growth rate in height for radishes, then the color of light would be my independent variable. It's what I think is responsible. Notice that the independent variable is on the x-axis of the graph. That's really important. The dependent variable is the variable that is potentially changing as a result of changes in the independent variable. So in this case, that would be here the height or average growth rate here. So we think this changes in response to this, okay? And notice that our dependent variable is on the y-axis, okay? So this is what we think causes the difference. Here is the result of the difference, okay? So independent, dependent. Controlled variables are things that we're going to make sure that we keep the same for all of our trials and treatment levels. So obviously, in order to get this data, someone had to expose radishes to 1, 2, 3, 4, 5 different colors of light. And I'm going to guess, since they found an average, that they had multiple trials of each. They probably, if they're good IB students, had at least 5 plants or 5 trials. Okay, so 5 plants, and they did each treatment level or color of light for these 5 trials. So we call this a 5 by 5, okay, that there are many different treatment levels, and they probably had at least 5 plants or trials for each of these treatment levels. Well, controlled variables are things that we make sure we keep the same for all of them. So if the light, okay, is the only variable we want to be different, then our controlled variables would be things like, okay, they all need to be in the same amount of sunlight. They all need to have the same soil. They all need to be the same species of radish. They all need to be exposed to the same watering routine. Okay, so those are our controlled variables. Okay, so another thing that your book doesn't do a great job of touching on here in the beginning are measurement types. And we're going to be using the metric system pretty exclusively. So let's do a quick review there. For length, okay, we're going to be using the meter as the standard unit. Okay, that's metric, and we abbreviate that with an M. For volume, that is the liter, an L. For mass, that is the gram, or G. And for temperature, we're going to be using Celsius, or capital C. Now, this is probably going to be something that you see and forget, but trust me, later on, or when we're talking about cell sizes, you're going to want this stuff to reference. Okay, so let's go ahead and list, okay, our prefixes and the number in a standard unit. And I'm just going to use meters as an example, but these could apply to grams, kilograms, whatever, milliliters or liters, sorry. It doesn't matter. Okay, so the first prefix we need to know is kilo. Okay, and there is 0.001 of these in a standard unit. So what does that mean? Well, that means if I'm talking about a meter, that means 1 meter is equal to 0.001 kilometers. Well, what does that mean? Okay, well that means that kilometers are 1,000 times bigger than meters. Okay, more on that in a moment. The next thing that you need to know is just the standard unit. So if there's no prefix, we're talking just the standard unit, so that would obviously be 1. So in other words, 1 meter equals 1 meter. Okay, there's no prefix there. It's all by itself. The next one down that we're going to use relatively often is centi. Okay, and centimeters or things like that, there are 100 of these guys in a standard unit. So 1 meter is equal to 100 centimeters. Now, next one down is milli. So these are big, and we're getting smaller and smaller as we go down. Okay, in a milli situation, we're looking at units of 1,000. So 1 meter is 1,000 millimeters. In order to move from centimeters to millimeters, I'm adding a 0. So 1 centimeter would be 10 millimeters. 1 to 10, adding a 0. The next unit down is micro. Okay, and it's worth noting that we have abbreviations for these. Kilo is K, centi is C, milli is M. Micro is this, mu. Okay, with this weird tail looking thing. And there are 1 million micrometers or micrometers in a meter. So 1 with 6 zeros, 1 million. So 1 meter equals 1 million micrometers. And the last one that we're really going to use is nano. Nano is N, and there are 1 billion of these in a standard unit. So 1 with 9 zeros, 1 billion. Okay, and so 1 meter equals 1 billion nanometers. These guys are teeny tiny. Okay, so notice if I want to go from here to here, that's a difference of 3 zeros. Here to here is 3 zeros. Here to here is 1 0. That's because I'm skipping some units in here. But anyways, I would just keep this chart in handy. Nanometers are really small, 1 billion. Micrometers, 1 million. Milli, okay, like a millennium, 1,000. Centi, like 100 cents, 100. And kilo, those are really big units, 1,000th. Okay, this is the last thing. I promise. I forgot to put this in your notes, so please just add this to the end on the back page. Super important that you know how to calculate percent difference or percent change. Ask any former IB student. You're gonna have to look at a graph and calculate the percent difference. So you're going to need to know this formula. Percent change is the final amount minus the initial amount divided by the initial amount, and since this is a percent, we'll multiply by 100. So let's say I get a question like this. Calculate the percent change from 2009 to 2014. Okay, time to start plugging in some amounts here. Gotta find the final amount. Well, the final amount is going to be the end of this date range that they're giving me. So in 2014, I had 286 million people on the internet. So 286 minus the original amount. So according to this date range, that would be 2009, and that would be 81 million. Okay. 286 minus 81. Hold on, I'm getting my calculator out.
286 minus 81. That was dumb. I didn't need my calculator. It's 205. Okay, now I'm going to take that 205 and I'm going to divide it by my original amount. So divide by my original amount, which again was 81. 205 divided by 81 is 2.53. So 2.53. Now I'm going to multiply by 100, and that gives me 253% increase. Is that okay that it's more than 100? Of course. Okay, that just means it went up by more than 2 times its own value. So 253% increase. Okay, that is okay that it's more than 100. I would love for my pay to go up 253%. That means I'm making several times what I'm making now, but that's okay. So just keep this formula in mind. Okay, final minus initial divided by initial times 100. And we are all done with our discussion on math. That is the longest bit of notes you will do for me all year. So congratulations, and I'm looking forward to practicing that with you later on.